All right. Thanks for coming, everyone. Whole 4 people. It's okay.
Y'all are rock stars.
It's okay, you sent the email all day, maybe we have some late.
We did forget to send out the email, but it's okay.
We'll do better next time.
So first review session for Bio 586. And we're gonna talk about CML. Okay.
Oh, look at the transitions. So topics for today, we're going to do an introduction. We're going to talk about hemoptysis, poiesis, cytogenetics, cytogenetic basis of CML, and then disease pathology, signs and symptoms, and treatment. After that, we'll leave some room for questions. We can talk about coursework, the papers, questions, anything like that. All right, to start off, we'll talk about leukemia and its different types. So to get going, leukemia is a terminology that was created for cancer in blood cells. It has Greek roots, so anyone who's studied Latin or Greek will probably know that leuk means white and hemo means blood. So you just put those two words together. And it was discovered through observing high white blood cell counts. And so on the left, we have our normal blood. And then on the right, you can see the leukemia cells, which are the blue and the purple jelly things. They're enlarged, and there's a very high count of those. The kinds vary in cell lineage and maturity of cells. There are 4 different types. So acute leukemias, which is this darker orange over here. Those happen in more immature cells, whereas in mature cells, that would be a chronic leukemia.
Can I say something?
Go for it.
Fun fact, the word chronic comes from the Greek root chronos, which is the Greek representation / deity of time.
Oh, Kronos is a god, isn't it? Or
It's a representation of time.
No, you're good.
Yeah.
No, you're good.
And then-
I know so little.
And then acute, I think it's from Latin and it just means sharp. So it's like a fast thing.
Oh, is it cute? Is that a god? Is there a
No,
god?
it's just a word.
Okay. And then the other 2 are lymphocytic, they come from the lymphocytic lineage, and myelogenous, which come from myeloids, which is the bone marrow.
We'll also send the slides out, so feel free to like not take notes if you don't want to. You just absorb stuff.
Just absorb.
Absorb, yeah. Here's another representation of it. This might be what I would put on a note card or note card. What is Laura calling them? Open note.
I don't know.
This might be something you can put on an open note and it kind of encapsulates all that in one. All right,
CML is chronic.
Chronomylogenous.
from Kronos.
Gonna talk about the other ones or just CML?
For now, we're just gonna talk about CML. For the later parts of this chapter, you'll see ALL, but that's like a minor thing. Oh, and what I'm gonna talk about it here. Okay, so before we go here, hematopoietic lineages. So these are lineages that originate from multipotent hematopoietic stem cells. And stem cells differentiate into different lineages and split even farther into different kinds of cells according to functionality. The younger cells are larger and the more specific cells are smaller. And just for what I'm going to do right now, there are more than just the myeloblastic myelogenous cell lineage, but emphasis for the myelogenous lineage will be on that specific line. Yes.
Cells the ones in the bone marrow? Or those are
The
different?
hematopoietic cells, the stem cells? Yes. Okay.
Myeloblastic or myeloblastic myelogenous cell lineage, is that the one specific?
Sorry?
The myeloblastic myelogenous cell lineage? that's the
Yes. So we're gonna go here. Oh, that's funny. Okay. I don't know why it does that. It's stupid. Anyways, so we're gonna pardon my not great handwriting. But we're gonna start at the multipotent stem cell. And it's hematopoietic. Pretend that I finished the last breath. So it starts here, right? And we're gonna move on to two different lineages. So I'm on the very soon. The lymphocytic first lineage cell will be your lymphoid stem cell. And the first cell on the on the myelogenous side will guess what? The myeloid stem cell. So it's myeloid stem cell.
I'm making you do a lot because you write out what the lineage.
The myelogenous lineage and the lymphocytic lineage. You didn't. That's good.
The great, but I was also. You're rocking it.
Good.
Okay. You're brilliant.
Thanks. Okay, and then this, we're just gonna pretend that the rest of the lymphoid lineage doesn't exist, and it's gonna go straight into leukocytes or lymphocytes, sorry. Which are your B and T. You might have heard of those through antibodies. Right? And so here, your myeloid stem cell can go into three. Well, again, there are more. We're going to focus on these. So it can become a red blood cell. It can become a platelet. Or it can become a a promyelocyte. Okay. Everything good so far? Okay. Promyelocyte, it goes into, you guessed it, myelocyte. And from myelocyte, it goes into meta myelocyte. Oh, not I, Y. Okay. So also keep in mind, the farther differentiated we are, the smaller the cell. That's important for something I'm going to talk about in a second. And from metamyelocyte, then you are going to go either into your neutrophil, your eosinophil, and your basophil. And this is as far as I'm going to get with the drawing. And I'll come back to it in a second. Okay. And here. So what are they? I just did a little drawing, but you have the lymphocytic lineage and you have the myelogenous. Again, like I said, it's more complicated than the lines I went over. There are more lines. Okay. Andrew will take the stage again.
Fantastic. Cytogenetics. Thank you for the drawing. That was just Mozart quality. Studying genomic mutations at the chromosomal level. All right, so the main four types, if we've taken 151 and maybe 285, we would have talked about duplication, deletion, inversion, and translocation. And then these mutations happen in the transition between metaphase, where we have our two little spindles and the kinetochores in the middle and the chromosomes, and they get ripped apart during anaphase. All right, and then there's also some other ones. So silent, these are point mutations where just one nucleotide is changed. It's in that little red and highlighted box. Even though it's changed, this codon still reads for a valine. So that's a silent mutation because it has no impact on the rest of the protein sequence and the amino acids aren't impacted. However, a missense happens when again, a nucleotide is changed, it's just one being changed and an entire amino acid is changed. So this was initially proline and now it's threonine. A bigger impact one, so these are progressively impactful for the proteins folding, right? Each amino acid is very important for the way the protein folds. So changing just one amino acid totally changes the structure of the protein. Now, a nonsense mutation is when you have a changed nucleotide, just one, but now it's going to code for a stop codon. And that's really bad because then the protein will just get cut off there and you're in trouble because the full protein amino acid won't form. And then finally, there's the frame shift or the insertion or deletion where they add two nucleotides. And so what's a good way? Could you have a good analogy for this one? All right,
So-
go on.
Codons come in groups of 3, right? And so let's say you have 33 total amino acids in the specific sequence you're looking at. If you add an amino acid at position 13, all of a sudden that 11 codon is going to be changed, and you're going to shift the entire frame by one amino acid. And so whatever nucleotide is that you added will change the size of the protein, because now where a stop codon would have been will not be a stop codon necessarily, and then you're going to have to be reliant on the existence of a new stop codon later on in the nucleotide sequence.
Perfect, yeah. And I think the Zoom is like covering it up. But basically the removal of these two nucleotides, well, there's a million of them. Just the removal of those two have changed the entirety of the rest of the codons made. So it's a very impactful when you insert or delete nucleotides into the. With that, I'm gonna.
You lost me, Andrew. It's okay.
You guys are rock stars.
Goodbye. Alrighty, so here we are, cytogenic bases of CML. So here we're going to talk about translocation event 922 at Q bands 34 and 11. And we'll be talking about the Philadelphia chromosome and more. So who here knows what chromosomal bands are? Good, because they're irrelevant, doesn't matter. It's literally just a position on the lower arm of the chromosome that the genes found. Qs. So it's Q34 for chromosome 9 for BCR, and it's Q11 for ABL and chromosome 22. But you don't need to know that, it's fine. So first off, we got to talk about BCR. So breakpoint cluster region is a gene found on chromosome 22 for humans, and really we don't know what the protein that's expressed real functionality is. What science has shown is that it has some sort of serine and threonine kinase activity. And it's also believed to serve as a guanine exchange factor for the Rho family of GTPases. So what is a guanine exchange factor? Yes?
Is that when you have GTP and the G protein receptor, and you're trying to pick out the GTP for the keeping out the GDP for the G.
And it
for the
helps it.
GTP.
And it does that. Yep, good job. Okay, so BCR, a little less information than ABL. And here we are going to talk about ABL tyrosine kinase. So this is a non-receptor tyrosine kinase. And is a gene found in human chromosome 9. And it is essential in some very, very important pathways that are present in our cells. These can be, as the image shows, a JAK-STAT pathway. Research has shown that it has impact on the PI3K AKT pathway. That's the cell survival signal one. It has impact on the RAS RTK MAPK pathway, and that one is about proliferation in growth. So as you can see, it plays some very important roles in some very, very important pathways. Also, it has been shown that ABL has some functionality in DNA damage pathways. What does that mean? This means that ABL, once expressed, has to go into the nucleus. For you to go into the nucleus, what do you need? Exactly. So if you have an NLS, you can get imported into the nucleus via important. And we'll talk about that in just 1 second. Alrighty. So, ABL, as we just talked about, performs some very, very important functionalities in essential pathways. Evolutionarily, it has developed its own auto-inhibitory mechanism. And so, ABL has a N-terminal myristoyl group binding site that causes a couple different conformational changes. And so once it is bound to the N-terminal cap domain, then the N-terminal cap domain will cause a conformational shift, which will cause the cap domain to bind to the kinase domain. Okay? And so once that happens, that generates another conformational shift. And so that creates binding space for a SH2 domain to dock onto the kinase domain. Once the SH2 domain is docked, it causes a 90-degree helical shift on a couple of the alpha helices that are present. And that becomes a proline-rich region. What domain binds a proline-rich region? The SH3 domain. How convenient. And so the SH3 domain goes in there, boom, binds it. Oh, also important, you get asked this in a future problem set. What's an SH2 domain? Anybody? SH2 is a phosphotyrosine binding domain, and it stands for Sarc homology 2 domain. Sarc, SRC, like sarcoma, homology 2. And it binds phosphorylated tyrosines.
So SH2 is a phospho.
Tyrosine binding domain. And SH3 is a proline-rich region binding domain. PX16 or PX16, which literally stands for proline something something domain.
So it's proline rich.
Region binding domain.
So you have your myristoyl group, which then binds to the kinase domain.
No, your myristoyl group binds the cap domain.
Yes. Which causes then causes a conformational shift.
So then the cap binds the kinase domain.
Okay.
The kinase domain then causes a conformational shift, which allows space for the SH2 domain to bind. Once the SH2 domain is bound, there's a 90-degree shift on alpha helices, which then allows for the SH3 domain to bind because the helical breaks are prolines, and that becomes a proline-rich region. Why is proline a helix breaker? Anybody know?
It's very rigid, and so it's not flexible.
But why?
Its side group connects back to the.
Exactly. So it doesn't have electrons that are available for non-covalent bonding. And so once the docking of the SH3 domain happens, the kinase domain cannot be active. Okay, so again, sequence of nucleotides enable N-terminal cap domain that contain the auto-inhibitory functionality, because if you can't bind the myristoyl, you cannot start the sequence of conformational shifts. Good? Okay, so what happens with the translocation event? So, Philadelphia chromosome. It is a balanced translocation event between ABL and BCR genes. And it forms a shortened chromosome 22 with starting with the ABL gene from chromosome 9. And then that's where that starts on chromosome 22. So it is right after BCR's sequence on chromosome 22.
Balance mean in this?
Balance in this case is a translocation event where no genetic material is lost. That means that the side of the piece of the chromosome that got translocated away docks onto the other chromosome that's receiving it, and the material is changed in place, but it is not lost. Of course. Wow. So now we have a chromosome. Fusion gene. Remember we were talking about the myristoyl group and the conformational shifts and that sequence? Guess what? With the translocation event, there's no more cap domain. How convenient. No more cap domain. And so the translocation event leads to the chimeric formation of the BCR ABL1 gene. What's a chimeric gene? It's a gene that's caused through a translocation event or through a mutation event, and it is expressed. And so when this specific chimeric gene is expressed, you get the BCR ABL1 fusion protein. Okay? And so the structural modifications lead to the absence of the so important N-terminal cap domain. On this drawing, this purple balloon that you got here is BCR, and the rest is ABL. And this is the C-terminus for ABL. And that's the N-terminus for BCR. Any questions so far? Go. Ask.
Is the myristoyl group also gone or just the cap domain?
It can't bind. There's nowhere to bind. So it's not even there. Okay. BCR ABL fusion gene continued. So, wow, how convenient. So we have two BCR ABL1 proteins. BCR has this specific domain called the oligomerization domain that we talked about, the coil domain in class. And that's where the two fusion genes that were expressed will bind to each other. So they're like, hold hands, they'll be right next to each other. When this happens, the kinase domains are conveniently going to be directly next to each other. What's going to happen? Cross phosphorylation. Wow, so then they're going to be constantly activating each other. What did we say ABL did in the cell? What essential pathways? And so that means that you have an overactive crazy enzyme with a nuclear localization sequence that has free access to the nucleus.
One of the paper said that it couldn't localize.
Well, but it has been seen to. So, I mean, I haven't dug like that crazy into it, but from the papers I have read, it has.
Maybe that was the 2007 paper and they had it figured out.
I also could be wrong. But considering ABL has that, we could like say that it does. You could also say allegedly.
Even without that, it's a shitty
Terrible.
answer.
Okay, so Laura talks about the term oncogenic addiction that is also mentioned in the paper. So overactivation of BCR ABL, as we talked about, caused increased activity of its kinase domain. In cell lines where BCR ABL1 is present, it goes crazy. It starts activating everything in sight because it cannot be regulated by its own autoinhibitory mechanism. It doesn't have it. So once that starts happening, the cell's like, oh, I have this crazy guy. He's doing all the work for me. I don't need to do any work anymore. I'm just going to let him do his thing. And so in cells where BCR ABL is expressed, it has been seen that the other tyrosine kinases are not as active. And so oncogenic addiction phenotypically describes cell lines that are driven into apoptosis once you don't have the oncogene present anymore. So if the malignancy, in this case, it's not a malignant tumor because leukemias don't cause tumor. But if it is absent, then it drives itself into cell death, which is apoptosis. Any questions? Yes. If the cell is oncogenically addicted to it and it's gone, yes. Yes?
So apoptosis, the treatment for CML is based on the fact that these cells, these cancer cells are oncogenically addicted to BCR-ABL, and when you get rid of BCR-ABL, they don't have functional kinase. and
Not
That's why they die.
necessarily. The goal is really just to stop the kinase from doing its thing. And then
Specifically, is the apoptosis caused because of a lack of kinase activity?
yes.
Got it. Sorry, I poured a bunch.
No, it's okay. But yeah, so once it's not there anymore, or once it stops working, it causes cell death. Okay, CML. Before we move forward, I want to go back to our drawing. And so here we are. And we have all this going on. Once we have that overproliferative phenotype, what's going to happen is you're going to get all of these cells from the myelogenous line that start off all the way here as a myeloid stem cell to have overproduction. So they're all going to be like proliferating like crazy. And while they're proliferating like crazy, also going to be not dying because of mutations that are being accrued. And so we were talking about the cell survival pathway. You have two RTKs that need to be activated by a cell signal, by a cell survival signal. If ABL can just go in and activate PI3K without a survival signal, then you're going to continue to trigger cell survival and your cell is not going to die. What happens? You have overproduction. Platelets, you have overproduction of promyelocytes, you have overproduction of myelocytes, you have overproduction of metamyelocytes, and you have overproduction of all of your granulocytes. All of them, they're going to be overproliferative. And so it's a good way to think about concepts we have learned in other classes before, because not only it's something that's happening because of one specific mutation, but also you have to think about the different pathways we've studied and all of the different intricacies that go behind kinase activation. Okay. So I'm going to stop sharing here and go back to the computer. And we're going to do this and share. So, CML, signs and symptoms, phases, diagnosis, and treatments. So different phases. We'll be going over the different phases more in depth in the next review session because it's more important for the third paper. It's more of a public health thing. But here's the breakdown of what the three phases are like. So first you have chronic phase. It's the first stage. It's where 80 to 85% of patients are going to be found in. So the disease is highly asymptomatic at this point. So you don't experience any real symptoms. Most of the patients find out about their cancerous condition through routine checkups, and we'll talk about that in 1 second. And so functional and effective Gleevec therapy is present. We'll be talking about how Gleevec is activated in a second, but this is just a breakdown of the disease. And very important, less than 10% of these cells are blasts. We were talking about on the drawing that you have overproduction of all of the cells. Blasts, or myeloblasts for the myelogenous line, are very, very big cells. As we were talking about, the more differentiated you get, the smaller the cell is because the more specific its function is. And so less than 10% of the cells are blasts. Accelerated or intermediate phase, whatever you want to call it, they're the same thing. So this is where the beginning of Gleevec resistance starts. Patients start developing symptoms that are characteristic of other cancers, such as fatigue, loss of appetite, loss of weight, lot of sweat. And some more specific symptoms that we will cover later in later reviews are splenomegaly, which is enlarged spleens, and bone pain. And there's this very, very specific reason for those two things. So keep an eye out for those, write them down. And here, 10 to 19% of these cells are blasts. So you more than double the amount of blasts. Blast crisis. It's the last stage. It's the most advanced stage, and it has a very, very fatal prognosis. It has a high mortality rate. So like I was talking about, the other types of leukemia we would cover, we won't really cover it, but they're very similar in a lot of aspects. And this will be Philadelphia chromosome positive acute lymphoid leukemia, or ALL. Splenomegaly and bone pain continue, and your cells start being unable to differentiate into their more specific kinds the more mutations you have. And here, you're gonna have a number of blast cells that is greater than 20%. That shouldn't be happening.
The one that highlighting the lineage study, I thought it was impossible.
Just for myelogenous in the bone marrow. And in the spleen, but we'll cover that later. Okay. So signs and symptoms. Patients will be usually older than 40 years old. This is happening in more mature cells at the beginning, as it is a chronic leukemia. Okay, so like I said before, 80 to 85% of patients are going to be in chronic phase. Okay, and like I was talking about before too, the common symptoms are fatigue, loss of appetite, fever, weight loss, you name it. Very, very common cancer symptoms. And here, I'll just explain a little bit about the development of bone pain and stomach pain due to splenomegaly. So remember I was talking to you about the higher number of blast cells that start occurring. What does that mean? Your bone marrow is going to run out of space because it's not supposed to have that many cells. Because you have a high elevated count of these more mature cells in chronic phase and later on the less mature cells, these blood cells are going to start leaving the bone marrow and they're going to start getting deposited in the spleen. This is why you're going to have this enlarged spleen. This enlarged spleen is going to be filled with these different blood cells that are not supposed to be there. And so you're going to have spleens that are like 15 centimeters big. They're huge.
So bone pain is because there's the cells in the bone marrow too big.
I'm going to get to it. So spleen is huge, and you have a lot of blood cells in there. What starts happening? What's the name of this entire pathway that I was drawing for the cells being developed? Hematopoiesis, right? One of the topics. You're going to start getting extramedullary hematopoiesis in the spleen. So these cells are going to start proliferating outside of the bone marrow in the spleen. The more these cells are proliferating, the more mutations they're getting, the more they're unable to differentiate, the larger the spleen, because the larger the cells. Okay, bone pain, we were talking about just now. Bone pain happens because you have bone expansion. The bone marrow will not be able to fit / comport all of these cells that are there, so it's going to have to expand. With that, your bones are going to have to expand. And with that, you're not prepared for it, your bones are going to hurt. So that is why you get bone pain. Any questions?
I missed a bit of why are the blood cells in the spleen?
They get deposited there because they can't fit the bone marrow. Okay. So diagnosis. There are two main ways to diagnose CML. First one is a full or complete blood count, which is a CBC test. And the second one is fluorescence in situ hybridization or FISH. Funny name. So, CBCs. It's literally a very routine checkup exam. People get these like a lot. And so a lot of patients for CML go into the doctor, they're just fine. They get the CBC, all of a sudden, their platelet counts are elevated. And most importantly, their myeloid cells are elevated. So you'll see 200,000 plus counts of white blood cells when the normal, like the paper mentions, is around 6 to 10,000. So you are in a very, very elevated number of white blood cells. And that is a image from the paper that they covered. And so this test is the test that precedes FISH because you go in for a routine checkup, you get a whole bunch of different exams done. One of them is a full blood count. All of a sudden you have 100, 200,000 white blood cell counts. And they're like, oh, this is not good. And then they schedule you for a different type of test. And this is a fluorescence in situ hybridization. So this is a technique which is reliant on fluorescence microscopy. And you are able to target specific genetic sequences, which are going to be attached to chromogenic probes. These chromogenic probes are fluorescent, okay? And they'll be sequentially matched to the specific gene that you are looking for. Once you are in the lab and you have successfully tagged these genes, you shine the assay under UV light and you can see the fluorescence go to work and you can see where your specific tag gene will be. And you can think about a different variety of uses where this could be helpful. It is not only in detecting CML. You could think about sex-linked diseases. You could think about trisomy events. There's a whole variety of different possibilities for this type of technique. Yes.
Does it look at the DNA or specifically the chromosomal structures?
It could be, you could look at positioning for chromosomes or you could look at DNA too. So I took these from my histology professor's slides. At least that drawing. That is an image from our paper that we were asked questions on right there. So here's the process. You fix the sample, you choose the specific DNA sequence you want, you get a fluorescent probe attached to the end of the sequence, you go into the cell, and then you're able to image it through fluorescence microscopy. If you look over here, BCR is tagged with red, ABL is tagged with green. If you look specifically at chromosomes, right here, they're fine. But then you look over here, you see red and green. They're together. How do you know they're together? Because you were able to use the specific genetic sequence of the genes and they appear together.
So they would take like a blood sample and isolate the cells.
Well, every cell that's diploid in the body has two pairs, right? So it could be literally any cell.
Do they have to culture them for them to be in a specific phase or does it mean?
Well, it's between meta and ana. Because you need, if you're looking specifically for chromosome formation, it has to be like that. The cells also have to be prepared to be permeated because, you know, you have to get through the membrane, you have to get through the nucleus, like there's a whole process. Yeah. So you use like Triton X100 or something to like get the cells permeated and then the probe goes in. Well, it can be, you can look at like a whole variety of different things. It doesn't have to be just CML. Like trisomy 21, for example. Okay. So treatment. They were looking at various different compounds. And we know that the functionality of the kinase works through activation of this case specifically tyrosines. So they were looking for methods to inhibit the activation of these tyrosines. So they can be utilized as targeted therapy for different cancer types, and inhibition works for different tyrosine kinases, EGFR, VEGFR2, PEGFR, which is for GIST, which we talked about, which is gastrointestinal. I forget what ST, whatever it stands for. It's that cancer that is mentioned in the paper too. And ABLE. So, in 1980 or in some part of the decade, the scientist team at Ciba-Geigy, which is now Novartis, big pharma company, found out this molecule through high throughput chemical libraries. This is one of the questions in your problem set. What are these? It's a whole bunch of different molecules, and like Laura was talking about in class, you get the molecule that you're looking to inhibit. You get all these functional molecules that you think might work. She says you throw them at the wall and see what sticks. And that's basically what you do. You look at the binding capacities and the factors that they alter for all of these different molecules. You have some fancy machines these days that could do like thousands at a time now. But back then it was probably a lot more complicated. And so once they were satisfied with their research and they moved along to clinical trials, those said trials pointed at the absence of side effects in patients with use. And it also pointed at extremely positive results. And I would like to point out this 92 to 98% of cancer cell lines that were complete cytogenic responses. As Laura was talking about, their method of citing as complete cytogenic response meant looking at 20 cells and seeing if any of them had it.
No, we have like a trillion cells.
Yeah, and Gleevec is very effective at, inhibiting the tyrosine kinase activity. But you have dormant cell lines that don't go active for years, which is why patients that are in remission, once they stop taking Gleevec, can go right into relapse. You have a whole bunch of these dormant cell lines, and they can go active like at any moment. If you stop taking the inhibitor, once they're not dormant anymore, they're going to proliferate.
Is Gleevec and imatinib the same thing?
Yes, that's just the company name. It's like Tylenol and acetaminophen. So, Gleevec as a drug became FDA approved in 2001, or imatinib, if you wanted to say imatinib. So, how does Gleevec work? So, Gleevec inhibits BCR-ABL1 function by competitive allosteric inhibition. So the molecule is going to bind to the ATP binding site of the catalytic domain, or of the catalytic site of the kinase domain. John, what does that mean? Instead of having ATP bind, Gleevec's going to bind, the kinase is not going to be able to be activated. That's competitive allosteric inhibition. And so, like I just said, it prevents kinase activity in the inactive state. Or I hadn't said that, but that's how it functions. It only works for inactive BCR-ABL. This was asked in class, and I wanted to cover it because it might have not been cleared for everybody. So it is assumed that these kinases that are active will go inactive at some point for some specific reason. And then once they are inactive, then imatinib can go in there, find the ATP binding site, and prevent them from regaining activity. So it's not that the kinase inhibitor is going to go to like an active kinase and shut it down. It's going to have to be shut down naturally for it to go in and prevent it from being turned on again. And that's a drawing from Wikipedia. I looked at the source. The source said you don't need to source me, so that's why there's no source. He didn't even like write his full name. It was just like, I forget the name of the guy.
It's like, I just wanna make this.
I just want to help. So resistance to Gleevec. So over time, like we were talking about, large number of mutations can be accrued by the different cell lines. So patients in especially accelerated phase started developing resistance to Gleevec. Some mutations can affect the structural component of the kinase. Some mutations affect the folding of the kinase. But a commonly found, question 7, mutation is the infamous threonine 315 isoleucine mutation. What does that cause? Who knows? Say it. Why?
Isn't it?
Why?
I believe it makes it even more active all the
But
time.
why?
Is that because it improves the binding between ATP?
No.
No.
It's because once you substitute a polar phosphate receiving electron carrying threonine for a nonpolar isoleucine, you lose the binding capacity of Gleevec. So you are no longer able to form your non-covalent interactions to where Gleevec binds. And all of a sudden, you cannot bind anymore. Any questions about that?
The gradient isoleucine, you said it changes from the polar to the non-polar, so that interaction. Does that prevent ATP from binding and Gleevec from binding, or
Just Gleevec
so ATP can?
still bind, and it does, which is why once Gleevec stops working, the functionality of the fusion gene continues.
So does ATP, and I know they're obviously allosterically
Compatible.
competitive, but does that mean that within the binding pocket, they're still binding in slightly different ways of this ingredient?
You're losing affinity, right?
Huh?
You're losing affinity once the mutation happens for both.
For both?
Well, yeah, because I mean, ATP still needs non-covalent interactions to happen, right? And hydrolysis is occurring. But for some reason, it's probably because evolutionarily the kinase domain evolved to fit ATP better, that it can still bind. Any questions? No? Okay. So because of that, there's the development of second generation inhibitors. So these are small molecule inhibitors, just like Gleevec, that were developed with mutation accruement in mind. So there are different amino acid mutations that can happen. I listed a couple of the common ones for the second generation inhibitors. You don't need to know them. I just listed them because they exist. So these are there in order to combat Gleevec resistance in patients. So they have slightly different binding sites and binding compatibility with the binding pocket. And so the second generation ones that are most common are dasatinib, nilotinib, and bosutinib, but you also have a couple others. It has been shown through research that there are less survival benefits when compared to first generation inhibitor. Molecules still perform design functionality of inhibition, but at this point you're with a high level of mutations in the cells and it becomes harder to catch. Is this the last slide? Yes. Okay, so I wanted to talk about this for a second that I was just talking about. So we are talking about BCR-ABL and how it starts off with the cancer process. It is not that because BCR-ABL is there that this whole cancer thing is happening. You have tumor suppressor genes, you have all of these cells that can have modified genes to perform ludicrous functionality. BCR-ABL just happens to be specifically involved in a lot of these pathways that are important for cell proliferation, cell survival, that are well regulated, that once it goes crazy, you can no longer control. Does this mean BCR-ABL is the sole cause for this? Not necessarily, but it just causes a perfect storm of cells developing new and new mutations every time, and you not being able to counteract it with anything. It is a perfect storm of potential for cancer to happen because you have all of the checkbox that are required happening. You have a lack of inhibition of important pathways. You have a strong presence of proliferation. You have a strong presence of cell survival. The cells are growing and you don't have a way to control it. At some point, you start developing too many mutations that not even the inhibitors can counteract. So you're also going to be asked questions potentially about how do you combo this with something else like chemotherapy drug? Well, you could talk about how chemotherapy, like Taxol, holds microtubules. And so if you are targeting like a small molecule inhibitor like Gleevec, while you're using Taxol to prevent anaphase from happening, right? Then you're solving, quote unquote, the inhibition problem, and you're also causing programmed cell death for these cells that are stuck in anaphase, right? So that's a way of doing it. Problem is, most of the time, when you're at that stage, it's too late. There are too many mutations. You can't control all of them. It's something that you fight with for Gleevec, for dasatinib, for nilotinib, for all of these inhibitors. But like I was talking about, 80 to 85% of the people that have it are in chronic phase. Majority of those people don't even know they have it. They go to the doctor for a physical, they take a full blood count, you have cancer. So it is unexpected. It is something that not a lot of people are aware about. You say leukemia. I mean, I didn't know before this class that there were 4 types of leukemia, right? And so is it a causal factor? Yes. Is it the only factor? No. That's what I'm trying to get at. Yes.
Is it? It's just a random mutation, right? It's not like
What do you mean?
genetically like some other.
I don't know the answer to that. But I would assume it's random. I mean, translocation events happen all the time. That one just happens to have an oligomerization domain, and then a kinase domain that can just cross-phosphorylate forever. Right, that happens to be like very, very convenient for those 2 genes. But you have other translocation events that also cause cancer that aren't as specific as this one. Okay, so I'm gonna close the recording here. Thank you all for coming, and I'll hold on. More.